Hello, everyone. Welcome to Introduction to Biosciences. I'm Anna, a member of the UCL IGEM 2020 team. So our team is made up of 8 undergraduate members and 3 supervisors. All of us will be contributing to the 3 webinar sessions that we are having over the summer. So just a quick overview. We're going to have 3 sessions, with the first one today focusing on molecular bio, natural sciences, and immunology. And our second one will be focusing on biochemistry, and the third one will introduce biochemical engineering. So now let's meet the speakers for our webinar sessions. First, for webinar 1 today, we're going to have 3 undergraduate members, Stefan, Pedro, and Juliette, and our supervisor, Kenth. Our second session will be presented by 2 undergraduate members, Daniel and Oliver, and 1 PhD, Alex. The third webinar session will be focusing on biochemical engineering, and it's going to be introduced by me, my undergraduate teammates, Laide and Lee, as well as our supervisor, Stephanie. So in these 3 sessions, we're going to introduce the bio-related majors that we are studying. That includes biochemistry, molecular bio, biotech, biochemical engineering, immunology, and natural sciences subjects such as medical physics and neuroscience. We will go over the core modules in these majors and their applications, as well as future career potentials. We're also going to share our journeys into these majors and our research interests. So hope you guys all enjoy that. Now, please, let's welcome Stefan and Pedro to introduce molecular biology.
Greetings, everyone. This is Stefan.
This is Pedro.
We're going to be introducing you to molecular biology.
So, what is molecular biology? Molecular biology is the study of biological molecules such as proteins and DNA and their interactions. So down on the right here, you can see a small video player, and it's a computer-generated video of how certain proteins in our cells interact to transport important molecules. And so the next thing we're going to talk about is the central dogma, which is, quote unquote, the big idea in the world of molecular biology. And what it describes is how the information that produces our proteins that are in different cells that do different things is all encoded or stored in DNA, which is shown here at the top. And so DNA is in the nucleus of our cells, or in the brain, as we call it in our whole bodies. And so it's transcribed into RNA, which is a single-stranded molecule shown in orange. And because it's single-stranded, it makes it easier to leave the nucleus, and then it can be translated into the proteins that our cells need.
So, given that, what is it that we actually study in molecular biology? So, on the left here, you can see some of the core modules. These include structure and function of biomolecules, specific takes on how exactly molecules function at the molecular level, at the atomic level. However, molecular biology doesn't end there. When you take into account all of the possible interactions between these proteins, then complex systems emerge. For example, what we studied in dynamic cellular systems, you take a look at how cells communicate within each other to essentially create life. On the right here, you can see what was one of my projects this year, where we had a molecule that binds ATP, which is a molecule you might have heard of, and we engineered it to fluoresce when it binds. This happens when you attach fluorophores to the backbone, or the back side of it, I guess. And you can see that the place where we attach these fluorescent molecules that give out a signal that you can detect is shown in green here with arrows. All right, so how exactly do we apply our knowledge to solve specific problems? Well, on the right here, we have a very conveniently placed DNA schematic. So we're going to take that, and as Pedro told you, we're going to make it into mRNA, which is what happens in cells. This is the process of transcription, and then in order to translate it into protein, some magic must happen, and the ribosome must bind. So when it binds, it promotes tRNAs to produce a polypeptide chain, also called a nascent chain when it's being made. And what you see here is how this nascent chain is exiting a structure within the ribosome called the exit tunnel. And now the schematic might look quite simple, but you're surprised to find that in reality, it looks more like this. Maybe not with all the fancy colors, but it definitely is a really, really complicated structure.
Okay, so that is a bit more detail on the central dogma idea that we were just talking about before. And luckily, at different steps of the central dogma process, we can intervene using molecular biology tools along with chemistry to improve things for our benefit. So for example, in antibiotics, which you've probably heard of before, we can use molecules that will interrupt this system. So for example, in that ribosomal exit tunnel, we can make molecules that block this, so bacteria can no longer make proteins, so they can no longer survive because they need these proteins for essential things. And also access to food. So for example, in areas that have poor food access, we can increase crop yields, which means increase the amount of crops that we produce or food produced by increasing this process on the right of producing proteins. We can increase this in certain plants, allowing them to grow more with the resources that they have. And also, apart from very practical things like antibiotics and food access, we can research things such as the origins of life. And also, this will tell us about the possibilities of life forms in outer space. So it's not just super practical things you can study. There's also very existential and important, interesting questions.
So how exactly do we come to these applications? There's two major worlds in the field of molecular biology where you can sort of do actual practical work. My specific area of focus is the wet lab, where I actually work with my hands to do practical science, where you physically have to mix things together to observe reactions and to basically make conclusions based on what you observe. So it's practical science, it's in a lab, you do a lot of experiments, but the one downside is that it's really time consuming and it generates a lot of data. So what do we do with that data? That data is worked in an environment called the dry lab because it's mostly a computer that analyzes what has been produced. So this world is very computationally heavy. It's primarily coding and programming, and it includes a lot of what we call bioinformatics, or the study of biological information. And you have to find patterns there, and you have to derive conclusions based on data.
So when I was asked to comment on the applications of my major, I said that the fact that you're learning core details of two major sciences, biology and chemistry, gives you a really good grounding for the future. Because say, for example, you want to continue your studies into a master's or a PhD, maybe, you have these two areas of science that you're coming from. So yeah, the applications are definitely not just the real world applications, but in terms of your studies and your career applications, it's very, very useful.
So carrying on from what you can do with your education, whether it be focusing on computationally heavy work and analysis, or producing results for this analysis, in reality, of course, things overlap quite a bit, and you get to do some of both. And this is particularly useful and is something that is heavily sought after in the industry sector. So that is one potential career pathway that you could be taking, producing biologically relevant products, for example, in agriculture, GMOs, even producing pharmaceuticals. There is another pathway that you can take, and that's called that's the academic pathway, where you'd be researching and primarily finding out answers to important questions in order to carry our understanding of the molecular world forward. Another thing you could be taking is something that I'd like to call in the middle, I guess, and this is the SynBio startup world, or the biotech startup world, where you have a startup that has developed a concept from some sort of gained knowledge, mostly through from PhDs, and they capitalize upon it by producing a product that could be sold to, for example, a bigger company or be developed into a product.
So these aren't the only areas that you could go into using a molecular biology degree or major. Your skills that you learn in these degrees will also be looked for in different sectors. So, for example, the finance world or the consulting world, they will also look for the analytical skills that you learn in this major.
I guess it would be really interesting for us to share with you how we got into the field of molecular biology in the first place. So take an example. I have always been into small experimentations at home, and I used to nag my parents quite a lot when I was young to buy me these small experimental kits to just play around with at home. But don't get me wrong, that is definitely not the only way you can get into science, and it's not the only way that you can do well in science. In fact, I was actually pretty bad in my first few years at university. What I really, really like about this course is that it teaches you the inner mechanisms of life.
So in my case, I was slightly different. So there's absolutely no requirement for you to have previous or background experience in experimenting really early on. That's not a requirement. My interest actually came from reading a really interesting book that I can recommend about the origin of life. It's called The Vital Question by Nick Lane. And this got me really interested in the field of molecular biology. And then from this, here in the box I've put that there's so many real world applications that have the potential to improve life. So reading that book actually opened my eyes to this. And one example of this is this protein called green fluorescent protein on the right here, which is actually found just naturally in this species of jellyfish. And now we can use this to attach to different proteins we want to study. And then using microscopes, we can see where these proteins go within cells. So it actually tells us so much about how cells work. And yeah, in terms of negatives, I would say that the only negative is that in the first year, you don't have any choice modules. But I wouldn't let that put you off at all, really. It's just a requirement to know the background information to be able to go on in second and third year and choose modules that you'd like to study.
So that is actually a good point to work out from. There is, because of the diversity and sort of how intertwined the degree is and how many overlapping fields are within molecular biology and the sister fields of biochemistry and even biochemical engineering, there is a large possibility of change. For example, I started with biotech in first year, or biotechnology, and throughout the year, I just more and more realized that biotechnology isn't for me. And now, don't get me wrong, biotechnology is an amazing degree. It just wasn't my particular choice, and it wasn't something that I wanted to do. So I switched to molecular biology, which also allowed me to do more hands-on work. So it is possible to switch between courses in most universities, and as long as you have the general direction, as long as you really want to be in the field of molecular biology study, whether that would be through biochemistry or through the more applied biotechnology, change is possible.
So that was Stefan on whether the choice of major can change in this degree. And now I'm going to talk about changes in perception of the major. So in the first year, a lot of the modules were the fundamentals and general topics that you need to know. And towards the end of first year, I was really looking forward to studying more applied modules. So at the start of second year, when I got to choose these things, I just found it super, super interesting. And so it's good to keep in mind that your perception of the degree will change as you go on, because the flexibility of it will give you choices that you can make yourself. So that's something to think about as well.
And now more of our story. What exactly do we want to do with our future areas of interest and future research areas? So I am planning on going into cellular communication, and that is basically the way that that cells communicate and share information in order to essentially function. So in order for cells to live, a lot of processes must occur and a lot of information must be gathered and must be distributed about these processes within the cell so that the cell can organize its functions and gear it towards living. So this is what particularly fascinates me, and this is where I've chosen to angle my interest.
So for me, the way in which I'm hoping to steer my degree and possibly a master's following on to PhD is disease research along with synthetic biology, which is the topic of iGEM, so this competition that we're producing these webinars for.
So what is our career plan? I mean, you might have guessed that since I gave such a specific example of what I'm interested in, I would be going into a more research-focused PhD degree, and then I plan on potentially, from the knowledge I've gathered, developing a startup. So that is one possible route that you can take. That is my near future goals so far.
Yeah, so the route that I'm looking at taking is a master's in research, which will be a whole year's worth of a research project, and that will tell me if I would like to go on to a PhD or maybe on something else. So that's my current outlook.
So we could end with a little bit of advice when choosing a degree in a related field or in the field of molecular biology. What I had to sort of find out the hard way was that reading about what specific things are being taught in a degree, or not only taught, but what exactly are you going to be able to do in terms of the knowledge that you gain in your degree. They really matter to your overall experience and to basically your interests. You have to be smart about your choices, and this isn't saying it in a very bad in a bad way, as in you have to go through an incredibly huge amount of information in order to be sure what you want to do, but you do have to be informed. So it's good to read beforehand into what fields you'd like to focus on, because reading and just reading a little bit before will save you time in the end.
So I definitely agree with the advice Stefan's just given. And then one tip that I've got to try and carry this out, to try and do a bit more research about your different options, is to try to learn more about different bio-related majors. And one way to do this is by looking at, for example, looking at the major discoveries in each field. So by searching timeline or history of certain field into Google and finding the Wikipedia page, it will give you a really nice summary of the history of that field and where we're currently at now. And this will inform you if it's something you would like to be involved with. So now we're going to hand you over to Juliette, who's going to talk to you about the natural sciences degree and her specific choices that she's chosen.
So thank you, Pedro and Stefan, for this really good introduction to molecular biology. I'm now going to be talking to you guys about my own journey. So I'm going to be talking to you guys today about natural sciences. So natural sciences is, as you can see, a course which involves a broad range of different subjects. And this is not even the exhaustive list and not quite a realistic representation of what natural sciences entails. And just as an example of how interdisciplinary and how much choice you get in this course, I live with my flatmate is also studying natural sciences at UCL, and she studies earth sciences and biology, whereas I decided to go a completely different route and will be focusing on today the study of neuroscience and medical physics. So to start with, what is neuroscience? Some of you might not know exactly what this entails, but in a nutshell, neuroscience studies the development, the structure, and function of the nervous system. It basically is the study of which enables us to understand how we function, how our brain enables us to walk, to talk, to basically interact with our surroundings. And so you learn about the simple anatomy of the brain and also its function, and as well as normal functioning, you learn about the what happens when things go wrong in the nervous system, which lead to disorders. And the second bit of my course is about medical physics. So what is medical physics? It's the study of the physics involved in medical technology and medicine. So I put some examples here on the slides. In this course, you learn about the different technologies that are out there which enable you to record from the brain using, for example, EEG electrodes, as seen on the slide, or imaging, actual imaging of our nervous system, but also of our human body in general. And then you also learn about the physics of our physiology. So the example I have here is about learning to model how our blood vessels function in our human body, and this can be modeled using laws of physics, using fluid dynamics, and using these mathematical models to understand how blood cells flow through our vessels. So moving on, when combining these two subjects, there's some core modules that you learn in this course, sorry. And this is not an exhaustive list of what you can study here. But very briefly, you'll learn about the basic anatomy of the nervous system, so how the different parts of the brain function together, what are the different brain areas that exist, and then you can also go into more computational aspects of it, which will lead us onto some of the applications that I'll talk about. And the image here that you see is an image of a single neuron. So we saw that we could do imaging of our whole human body, but as well as doing that on a large scale, you can do imaging on a very small scale, looking at single neurons like you can see on the slide. So in terms of the applications of these two, when combining these two subjects together, and a strong application in neuroimaging, and an example of it here is PET. In the previous slides, you were looking at scans of MRI, and this is a different kind of scan, which is PET. And the basic principle of it is in basically injecting a radionuclide into the patient's body, and this radionuclide is basically enables you to visualize different like metabolism and activity levels in the body. And this is really useful for detecting cancer, as cancer cells tend to suck up a lot of the energy in your body, and this is very visible in a PET scan. As well as during these like very clinical related imaging, you can also do what I did over the summer last year in a neural computation lab, look at doing time averaged photos of a mouse's brain. And I just want to emphasize how much the lab I was working in emphasized the well-being of the animal, and how it was really the top priority of the researchers there. And working with these mice, you could get some very beautiful images of their brains. In this experiment, we were stimulating the mouse's whiskers and giving this sensory stimulation. You could see bright spots of activation in its brain, as you can see on this image. So as well as looking at images, neural computation is all about learning how neurons communicate between each other. And this is crucial for learning to decipher artificial intelligence, for example, which you will all know is a really hot topic nowadays. But I won't go into the details of that. And then finally, the final application is medical electronics, which, as we've seen before, enables you to record some brain signals in the brain. And there's also constantly a strive to improve these methods of recording. And this is what people usually work on in research. In terms of the careers you can go into after such a course, well, these are, again, a short list. There was as you could see, there was a broad range of applications. There's also a broad range of careers. But you can go into clinical work as a physician associate, which works really closely with doctors and radiologists. You can also obviously work in research, looking at various applications that we've seen in the previous slide. And here there's an example of doing microscopy work and staining neurons individually, as you can see here, where individual neurons are stained with different colors. depending on the properties they have. And this is an image of my own work when working in the lab last summer, where, as you can see, we injected a fluorescent dye through the brain, the mouse's brain, and very visually, it really shows up how the neurons communicate between each other, in what direction they do that, and it gives really beautiful images, which I think is fascinating. And then finally, you can work in industry, like most other bioscience, as Stefan pointed out. And an example here is this company called ThinkSono. So there is definitely possibility for change in natural sciences more than in any course, I'd say, because of this flexibility of choice. And so I actually started coming into uni thinking I would study biochemistry, and I actually discovered neuroscience and how it could be applied with physics, and that I really became passionate about that. So I was able to switch, and I'm really glad I did. So as a bit of an overview of what natural sciences is, it's really, big focus on the interdisciplinary aspect. It gives you loads of flexibility, and there's really an opportunity to expand your social network immensely. And this can, just as a more realistic point of view, this can also backfire, because it's possible to find you, like I've had before, finding myself in class and lectures where I don't know anyone because I'm coming in from a different course and studying these different modules. And in as natural scientists, we're all very spread out across different disciplines. But this shouldn't be something to discourage you. It's more, I think, an opportunity to then expand your network even more, to kind of gain in confidence. And I've definitely had that advantage from studying natural sciences. And more about what I'm more interested in terms of research. My actual area of research at the moment is brain-computer interfaces, which I've worked on a lot this year. And this is actually, for those of you who don't know, the basic principle of it is the aim is to help people who are disabled, who can't walk anymore, who can't speak, to be able to function properly and normally again. And this is done by recording brain signals, by processing them and giving them to a computer or a machine to give a command. So this is the case for when controlling prosthesis, for example, just by using mental power and simple brain thoughts to control a machine. And I think it's really interesting and fascinating field of research. And what really got me into this field is this first study that I was working on in one of my modules for biophysics, which was showing this woman who was a tetraplegic woman, and she couldn't walk anymore, couldn't make any movements, and they trained a brain-computer interface on her. And this image shows her finally being able to give her, like, eat chocolate and feed herself chocolate. And it really, you can see that it really sparks joy to be able to do something like that again. And I think that's what really pushed me to want to spark joy in people again. So very briefly, my career plan is very similar to the things that I've talked about before in terms of what careers you can do after this degree. And I'm really interested in working in AI and artificial intelligence, potentially in a biotech company. And then I would also like to go back to research at some point, maybe undertake a PhD, but this is all very unclear. And this really leads me on to my final point, which is it's kind of okay not to know what you want to study. As you can see, I don't even really know what I want to do after my degree, and I'm already a third year. So this is, you must think, well, isn't this, I need to choose soon. I have about 6 months to really get my head around that, and that's okay. So don't be afraid of not knowing what you want to study, and it's okay to make mistakes, to try again. So yeah, I just want to give a final point of advice of not giving up and trying to study what really makes you the happiest. So yeah, thank you. And now we will have Professor Kenth to talk to you guys about immunology. Thank you.
So, hello, my name is Kenth Gustafsson, and I'm going to talk to you about immunology, that is the immune system in our bodies, which is of course there to fight infections caused by bacteria, virus, parasites, etc. But of course the immune system is more than that, and also involves a number of different diseases, which I'll talk a little bit about later on. We in our group are interested in how T cells work with T lymphocytes. So T lymphocytes is one cell type in the immune system, and they can kill cancer cells on a good day. They can also direct how we kill bacteria and virus-infected cells. We're also interested in a certain type of antibodies that are secreted from B cells, the other type of lymphocyte in the immune system. And particularly the antibodies we're interested in are called natural antibodies that control glycosylation. And actually, they also interact with viruses in that they interact with the carbohydrates or glycosylations on virus. So, as an example of what the immune system is involved in, perhaps a good idea to talk about COVID-19, which is the awful epidemic, or pandemic, I should say, that we've all been more or less affected by over the last few months. Like many other viruses, COVID-19 is consisting of a number of different genes. So they have genes like we do. They have structural genes, in the case of COVID-19, 4 genes that encode proteins that help to maintain the actual structure of the virus. But other genes code for non-structural proteins in the virus that can try to fight our immune system, stop our immune system from trying to get rid of the COVID-19 virus. And then there are some other genes as well. It's actually 29 genes in the COVID-19 virus. And by comparison, of course, we have around 20,000 genes, so they have much fewer than us. One of those proteins coded for by one of the structural genes making a structural protein is the spike protein, and that's the spike protein that attaches the virus to a receptor on our cells, which is the beginning of the process for COVID-19 to make its way into our cells. And it's a particular domain on the spike protein called the receptor binding domain, or RBD, that is responsible for attaching to our cells. And when it does bind to the receptor called ACE2 on our cells, to begin with epithelial cells in our throats or in our noses, that is how the virus then makes its way into the cells, where it can then start to replicate its RNA genome, where the genes are. So that it can make more viruses that will then exit that cell to go away and infect other cells in our body or indeed in someone else. So what's happening to the COVID-19 pandemic? Well, there is a lot to say, but I'm going to mention as an example a potentially successful treatment that has just come about only a week or two ago here in the UK at Oxford University. One of the major problems with COVID-19 is that it can cause, as you know, people to be very ill indeed. Fortunately, only a minority of people, but still ending up in intensive care. And what they suffer from is something called cytokine storm. So actually it's not the virus, paradoxically, that kills people, but it's rather an overreaction of our immune systems that results in death, it is believed. And what happens is that the signaling molecules that are sent between cells to help cells within the immune system to communicate, like people talked about earlier in the previous presentations. And within the immune system, those communication molecules are called interleukins, or cytokines. So that's where the name cytokine storm comes from. And in this disease, it's in particular IL-1 and IL-6 and a couple of other cytokines that are overproduced. And this can lead to a complete overreaction of the immune system. What the people in Oxford have recently found is that a rather common medicine called a steroid, like dexamethasone, can actually be great help to the very ill patients in intensive care. This is still under clinical trial, but it's looking very promising, so that a number of people in intensive care can probably be helped by this simple and very inexpensive medicine. But of course, the ultimate solution for COVID-19 that we're all hoping is coming along as soon as ever possible would be a vaccine, an efficacious and efficient vaccine. And a vaccine can work in two different ways, and ideally work both those ways. One is to instruct the production of antibodies that are specific for the virus, so that we can bind our specific antibodies in our bodies to the virus and thereby help to destroy the virus. In addition, ideally, a vaccine should also trigger T-cell responses to the virus, which are also highly specific. So those are the aims of an efficient vaccine. So we can all just hope that one comes along soon. In addition, the involvement of immunology in COVID-19 also touches on testing for COVID-19. And that is when one wants to test whether someone has had COVID-19 previously. But first, in contrast, if someone has an active infection with COVID-19, that does not involve testing the immune system or aspects thereof, but rather just testing using polymerase chain reaction if the individual has viral RNA at that particular time or not. But if you want to test if someone has had the virus previously, then one way is to look at if that person still has antibodies against the virus in blood, and hopefully one day soon in saliva as well, using a fairly simple slide similar to a pregnancy test. So, more broadly, what is immunology and what is it about? When you study immunology, when you read immunology at university and maybe choose to enter a career in immunology, well, it's a branch, it's a part of biomedical sciences. So by that and because of that, it also contains a lot of other subject areas within it, such as biochemistry, genetics, molecular biology, cell biology. And it's basically a question of studying the structure as well as the function of different aspects of our defenses against infectious diseases. But as I said before, it also deals with other issues other than infections. So apart from vaccine development, it studies immunodeficiency disorders, which is something you can be born with, autoimmune diseases, some of the most common diseases in humans, like rheumatoid arthritis, diabetes, multiple sclerosis, where we start to attack our own or parts of our own bodies. Allergies, of course, but then also transplantation of either cells or whole organs from one donor to a recipient. And what happens there is that the recipient of tissues or organs from another donor recognizes the new organ or the cells coming into the body as if it was a foreign cell, which is of course the idea with the immune system. So what careers can you enter into within immunology? Well, immunology can, of course, be very clinical. So as having studied medicine, for example, and you're a medical doctor, you can specialize in immunology. Or you can be a biomedical scientist, in which case you perhaps start with biology or biochemistry at university and you then specialize towards immunology. Or you can study pharmacology and then, as a subspecialty of pharmacology, you can then study immunology. Because after all, many medicines are intended to modulate the immune system. And again, as I said previously, vaccines are part of that. You can be a molecular biologist. You study the molecular functions within the immune system in an academic setting at a university, or indeed, of course, in industry, pharmaceutical industry, as I said before, or in biotech industry.
So thank you, Professor Kenth, for your presentation on immunology and your advice to our high school students. And finally, thanks everyone for attending webinar 1. We really appreciate the support from all high school teachers and students. A quote we'd like to share with you all: "Everything is theoretically impossible until it is done." We really wish that everyone keeps pursuing your passion in biology, and looking forward to seeing you in webinar 2.